Okay, so good morning, everyone. Still good morning, yes, and Merry Christmas, everyone, as well. I just wanted to give a bit of an overview of some of the work that we've been doing in the lab here, and specifically the work that Debbie has completed over the last 4 years. And as some of you will have seen, Debbie has now finished her PhD and moved on to Pastors New at the NICB. So I just wanted to give everybody a quick overview of the conclusions from Debbie's work, and also then just kind of highlight some of the aspects that potentially you could collaborate with the lab and some of the work that you're doing here in the School of Nursing. So just want to outline the project is based around prostate cancer. And prostate cancer, while it can be a slow growing cancer, which just requires some monitoring, and the patient can, in certain cases, can outlive the disease, there is a cohort of patients who have more advanced or aggressive disease. And as I've outlined in the introduction, this more aggressive disease, the gold standard treatment for that is androgen deprivation therapy. So essentially, androgens are the growth factor for prostate cancer. And by removing those androgens, that can help slow the growth of the cancer, of the tumour. However, there's a significant amount of resistance associated with this treatment. So typically after about 2 years, a patient will become what is termed castrate resistant. So they're no longer sensitive to androgens, and the prostate tumour will continue to grow whether androgens are present or not, and that is associated with patient mortality. So there's really an unmet need for new therapeutics for castrate-resistant prostate cancer, and that is where this project comes in, trying to identify new targets that are associated with this resistant disease, and that, if we can identify new targets, could improve the overall survival of patients. In relation to this project and in relation to a lot of different cancers, there's various different genomic alterations that drive cancers and tumor growth. And in prostate cancer, there's one particular alteration called TMPRSS2 ERG, which is a bit of a mouthful, but it's a fusion gene, and it's found in approximately 50% of cases of prostate cancer. And why that is important will become apparent as we go through. And the other aspect then it's important to know about this project is it involves and investigates calcium signaling. So calcium, it is calcium that you find in your milk, that metal ion. It is a vitally important ion that is found throughout your cells and supports the normal functioning of your cells, supports proliferation, gene expression, various different aspects. And therefore, considering it plays such an important role in the cell, it's not, I suppose, surprising to find that aberrant calcium signaling plays a role in disease, in various different diseases. So not just prostate cancer, but other diseases such as Alzheimer's and so on and so forth. It has a big role in disease generally. So here we're going to look at calcium signaling and its role in prostate cancer. And particularly, we're going to focus on one calcium channel called CAV 1.3. So this calcium channel allows the passage of calcium into the cell, and it controls various different aspects. And this calcium channel is known to be regulated by ERG, which, as I mentioned earlier, is this genomic alteration that's present in about 50% of prostate cancer patients. So our overall aim was really to investigate the impact of this channel on intracellular calcium in prostate and see if it was able to drive progression to castrate-resistant prostate cancer. So there was a number of studies that highlighted CAV was upregulated in prostate cancer, but it didn't go into any detail, nor did it explore its role in this more terminal castrate-resistant prostate cancer. So our first port of call was to conduct a bioinformatics study, looking for the expression of this channel in prostate cancer patients to see if there was any trends of increased expression or any association with the more terminal resistant cancer. And I apologize, this is quite small here, if I can zoom in. There we go. So in figure 1 here, and the figure legends are quite difficult to read, but I will talk you through it. So in figure 1A, the blue box here represents normal samples, so non-cancerous samples. The green represents primary tumor, so an early stage tumor, and the red represents metastatic castrate-resistant prostate cancer, so that terminal prostate cancer. And each of these boxes is measuring the expression of this calcium channel. And you can see quite clearly that as the disease progresses from a primary stage to a terminal stage, there's a significant increase in this calcium channel. So that was very interesting for us. And not only that, that we found here in figure B, that androgen deprivation therapy, which is the main treatment for prostate cancer, in those in the blue box here that haven't received any, the expression is much lower than those who have received androgen deprivation therapy. So it suggests then that this calcium channel is increased during this treatment and is potentially driving the resistance to that particular treatment, which is a very important result. Not only that, but you can see here in this figure that we looked at ERG expression and the association of CAV with that. And we find that those who are ERG positive have that genomic alteration that's quite common in prostate cancer. They have a significant increase in CAV 1.3, as you can see in the green, compared to the red here. So again, we know that this calcium channel is present in a large cohort of patients, and specifically patients carrying this genomic alteration. And that's particularly important discovery for many different reasons. One being that the way we target and treat cancer is changing, that we're moving to a personalized medicine approach, and we will go about treating cancer patients by looking at their genomics and identifying patients with particular genomic alterations like here, and giving them specific treatment rather than just a blanket treatment of chemotherapy or surgery or radiation, for example. So very exciting results with that. And also we find that the expression of this channel correlates with Gleason score, which is the score used to give an indication as to the progression and how aggressive the disease is. And then lastly, it is associated with the probability of biochemical reoccurrence, i.e. androgen deprivation resistance. And you can see going from blue to the light green, the higher the expression, then the shorter the time until biochemical reoccurrence. So again, reinforcing that CAV 1.3 drives resistance to androgen deprivation therapy and therefore promotes this terminal form of cancer. So that was very important information, and it tells us that CAV plays a role, and we wanted to then investigate what role exactly CAV is playing in disease. So this was in patient samples using bioinformatic methods. So therefore we moved to the lab to more specifically look at CAV in a cell line model and try and understand what exactly this channel is doing. So when we looked at the expression, we developed our own cell line model of androgen deprivation therapy and castrate resistance. And we can see again here in figure 4B that there's a general stepwise increase as we go from initial androgen treatment to castrate resistance. I'm going to skip over figure 5 here in terms of time, but we also repeated that in a mouse model and achieved the same results. And what exactly CAV is doing, we can see this in figure 6. So this figure 6 is a calcium trace. So we took our cell lines and measured the levels of calcium, intracellular calcium inside the cell. And this would give us this trace over time, as you can see. And the important part here is that the end of this trace. So the black line here represents a cell line that is androgen sensitive, so it hasn't been treated by androgen deprivation therapy. And the red line gives us a calcium trace for cells that have been treated with androgen deprivation therapy and have developed resistance. And you can see the red line is much higher than the black line, and that indicates then that there's more calcium coming into these cells, and that correlates with the increase in CAV 1.3. So we know CAV is increased in prostate cancer due to resistance of this treatment, and we know that it's transporting calcium. So can we target that, and can that have a beneficial effect on prostate cancer? So that is what we finished off with. So looking at proliferation and survival of these cells. And we saw then, if we look across various different prostate cancer cells and we target CAV 1.3 with a knockdown approach, you can see with these knockdown here, the proliferation significantly decreased. And also then we looked at cell survival using an existing drug called nifedipine. So nifedipine targets and inhibits CAV 1.3. And when we use that existing treatment, that again significantly reduced the survival of these prostate cancer cells. So this project really gives us a big increase and an important discovery in terms of personalized medicine approaches for prostate cancer, and it identifies this calcium channel as a key target in prostate cancer progression and treatment resistance. So I know there's maybe some unanswered questions there, and I don't want to go into too much detail with the time. So I'm happy for further discussions afterwards. But the other thing I just wanted to draw everybody's attention to was there was recently a grant call from the Irish Research Council looking for combined grants between human science and more basic science, like the work that we do in the lab here. And I think there could be a number of significant overlap with my work and others, even utilizing the lab to analyze samples from some of the work that you're doing. So I'd be very interested to talk to people who are interested in cancer or interested in investigating some of the basic science aspects of your work as well. So thanks for listening. I'm happy to take any questions around that.